All right, hey everyone, welcome back to the Viking Initiative for Building Excellence. This is IB Bio, and today we're going to be talking about biomolecules. So, the first important thing to know is that the element carbon is in all the biomolecules, it's the main element in all of them. And the reason why it's important in biomolecules it's because it's able to form full covalent bonds, meaning it's very diverse and it has many wide uses, as is necessary in biology. So, an important concept to know with all biomolecules are the way they bond. And there's two types of chemical bonding that goes on in biomolecules. First is condensation, and second is hydrolysis. So, in condensation, what's happening is removal of a water molecule to form a polymer. So, in this picture, you see on the left side, there's an arrow pointing outward, signaling that water is being removed. And this is coming from the OH group on the left side and the H group on the right side. And this forms a carbon bond that links these two, forming a polymer. On the right side, you see that there is an arrow going in to this reaction with hydrogen, water. So it's coming with an OH group and an H group. And this is splitting the carbon bond and adding an OH group to the left and a hydrogen group to the right. And this is forming two separate monomers. Okay, so we're going to start with carbohydrates. Um, the monomer of carbohydrates are monosaccharides, and there are two types of monosaccharides necessary to know. The first type are pentose sugars, and the second are hexose. So for pentose, some examples are ribose and deoxyribose, and you would find these in RNA and DNA. And for hexose, glucose, galactose, and fructose are three examples. So you can see that glucose and galactose, they have a hexagonal shape, and deoxyribose and ribose have a pentagonal. Fructose also has a pentagonal, but you can see that there's two CH2OH groups, meaning that it has six carbons. And next, we can look at some disaccharides. So we're going to look at the top three. So if you combine glucose with another glucose using condensation, you'll form maltose. Glucose and galactose makes lactose, which is the sugar found in milk. And glucose and fructose is sucrose, which is common table sugar. Um, the most important one out of these is glucose, and it's important to know that it is soluble, hydrophilic, and polar, and this is in contrast to lipids that we'll find later. It's very chemically stable. It's able to be oxidized, which is important in cellular respiration. And there are two types of glucose, alpha glucose and beta glucose, which we will look at in further comparison. Okay, so another thing to know is that each one of these biomolecules has covalent bonds and they all have different names. So in carbs, they're called glycosidic bonds. So if you see the OH group on the left and the HO group on the right, imagine removing an OH and an H, which equals a water, and you would form that C to O bond, um, forming the disaccharide. And in this case, this is maltose. So that is a glycosidic bond. So we're going to look at some different types of carbs. The first is starch, then glycogen, then cellulose. So the two types of starches are amylose and amylopectin. Both of these are found in plants. Amylose is more simple than amylopectin. It is linear, whereas amylopectin is branched. And amylose only forms 1 to 4 bonds. And what this means is that the way you count glucose is based on its carbons. So this is formed between the first carbon and the fourth carbon, and amylopectin forms between the first and fourth and first and sixth carbon. This creates the branch structure. And they both utilize alpha glucose. Similarly, glycogen is also an energy source, however, this is used in animals. It is also branched like amylopectin and it has the same 1,4,1,6 glycosidic bonds. It also uses alpha glucose. Now, kind of the outlier here is cellulose. It is in plants, but it is used for structure. It makes up the cell walls of plants. Um, the structure of these is not linear or branched, they're in sheets. Each sheet is held together by hydrogen bonds or hydrogen interactions. So these are not bonds per se, but just sort of interactions that are called hydrogen bonds. And these only have 1,4 glycosidic bonds. It is more simple and they're stacked up in sheets, but these are made up of beta glucose instead. And here is a picture of all of them just for reference. So this first one right here, a starch that is unbranched, that would be amylose. A starch that is branched is amylopectin. We have glycogen. It is also branched. It is more branched than amylopectin, and cellulose is in sheets. The last thing to note with carbohydrates is glycoproteins. So these are found on the cell membrane, and they have a role in cell-to-cell recognition. They can act as receptors. They can act as ligands, which bind to certain receptors, and they contribute to the structure of the cell membrane. And one important example of these are in blood type. So we have A, B, and O blood types, different combinations of each. And the way these come about is that people who have A blood type have the A antigen, and these antigens are glycoproteins. So these proteins differentiate between the different blood types. Okay, next is lipids. So these, in contrast, are insoluble, hydrophobic, and nonpolar. Um, the fatty acid is the monomer of lipids, and it's composed of a carboxyl group plus the chain of just the hydrocarbon chain and a methyl group. So that methyl group is just a cap on the chain, which is the CH3 molecule. And if we look at two types of lipids that are important to know, we have triglycerides and phospholipids. The way these differ are triglycerides are composed of three fatty acids plus a glycerol backbone, that's picture A, and phospholipids are composed of two fatty acids, glycerol backbone, phosphate group, and a head group, which sort of differs. Okay, so the covalent bonds in these are called ester bonds. If we look at this glycerol group plus a fatty acid, imagine you're taking the H group and the OH group out, then you're left with the O to C bond, and that's called the ester bond right there. This is polymerization or condensation reaction. So we have some different types of lipids. First is saturated, and the way I like to remember this is SSS. So we think of this as single, straight, and solid. They have single bonds only, they're straight, not kinked, and they're solid at room temperature. For unsaturated, we have two types, so cis and trans. Cis are the ones that you'll find naturally. There's two types of cis, which are monounsaturated and polyunsaturated. All this means is that mono has one double bond and poly has multiple double bond, and this correlates to one kink and multiple kinks respectively. And they're both liquid at room temperature. And if we look at trans, these can have one or multiple double bonds, but the way it differs is that the hydrogen are on alternate sides, which eliminates the kink. So it does have the double bond that a mono and poly unsaturated would have, but it is not kinked. Okay, now if we look at proteins, the monomer protein is an amino acid. So the way these are composed of is this central carbon. You have this hydrogen off the carbon. The amine group is this NH3 on the left. The carboxyl group is COOH on the right, and the R group is the variable. So there are 9 essential amino acids, meaning that we cannot get them, we cannot synthesize them ourselves, so they have to come from food. So if we look at the covalent bonds in proteins, they are called peptide bonds. Right, so imagine this OH from amino acid 1 and the H from amino acid 2 are being removed, then you'll see the C to N bond, and the H will flip up, and that's called a peptide bond with the removal of water. That's the condensation reaction here. If we look at proteins, so there are 4 different types of structures of proteins. The main 3 are primary, secondary, and tertiary. Primary structure is simply the polypeptide chain. So this is just the amino acids and their peptide bonds. Very simply, just a chain of amino acids, as you can see in the first picture. Secondary structure, um, there's hydrogen bonds forming between the different amino acids, and this can form either uh, pleated sheets or helices, which you can see in the second picture. Now, tertiary structure is more complicated. This is the interaction between the R groups, the variable groups. So there's 4 types of interactions. The first is hydrogen bonds, then disulfide bridges. So this occurs with R groups that have sulfur. Ionic bonds occur between charged R groups, so they're ionic interactions, so positively, negatively charged R groups attract each other. And then hydrophobic interactions between nonpolar R groups. And these just increase in complexity the structure of the protein. Lastly, we have quaternary structure. So this can be multiple tertiary structures put together. As you can see, there in this left picture of in the right picture of hemoglobin, there are four different chains here. You can also have something called conjugation optionally, which is the addition of non-amino acid elements. So in the case of hemoglobin, there are heme groups added, which are composed of iron. Um, in the case of collagen on the left, this is a non-conjugated protein, meaning that it is just a combination of tertiary structures without the addition of non-amino acid elements. Now, this can be also divided into globular and fibrous proteins, and this is just based on the shape. Fibrous is going to be more, lengthwise, more straight and simpler, um, and globular proteins are going to be like more like a glob, like enzymes. And lastly, we're going to look at nucleic acids. So, the building block of a nucleotide is the building block of a nucleic acid is a nucleotide, and it is composed of a pentose sugar, right? So either ribose for RNA or deoxyribose for DNA. That same phosphate group, so PO4 2 minus, and that nitrogenous base, and there are four different ones. Okay, so the covalent bonds in this are phosphodiester bonds, so they all have a different name. And it's important to note that the 5' end is the phosphate group, so you can kind of imagine it like this little circle, it's waving high, right? With all five of its fingers. If it was a person, that's how you can remember it is the 5' end. It's waving high. And the 3' end is obviously the opposite end where this 3 carbon is. And one thing to know is that you can only add nucleotides to the 3' end. An important concept of DNA is that it runs anti-parallel, meaning that if you draw this structure with the pentagon for the pentose sugar, the little circle on the left, and the nitrogenous base on the right, you draw those going all the way down with these phosphodiester bonds in between. Then, if you were to turn your paper upside down and draw the same thing, it would be perfect because there would be anti-parallel. So one side is running 5 to 3, the other side is running 3 to 5. Okay, so just to compare and contrast DNA and RNA, DNA is double-stranded while RNA is single-stranded. The purines in DNA are adenine and guanine, and it's the same for RNA. And the pyrimidines in DNA are thymine and cytosine, whereas the pyrimidines in RNA are uracil and cytosine. So pyrimidines and purines are the nitrogenous bases, and it's important to note that a purine always binds to a pyrimidine. Um, so for DNA, it's TA, C, and G, and for RNA, it's UA, C, and G. Um, the last thing to note is a nucleosome. So this is the building block of a chromosome, and it consists of a DNA wrapped around 8 histone proteins for protection. And this is linked to each other by linker DNA and stabilized by the H1 histone protein. Okay, actually, before I leave off, just one thing to note is that with all of these biomolecules, it's very important that you know how to draw the structures of all of them and you understand the form as it relates to its function. Um, yeah, that's it. Thank you so much for watching. Make sure to visit the Viking Initiative website for more APIB resources. If you have any questions, you feel free to join the IB Bio group chat and you can ask us. Thank you for watching.